There you go.
All right, and then if I want to talk to him, I need to.
Leave it muted.
Okay. Can you hear me, Gregor?
Yes, very well.
Great. All right. So, thank you everybody for your patience. It's quite a challenge because we do have an international consortium, and so some of our scientists and clinicians are logged in all over the world here for this, and it's really my pleasure to be giving this presentation with Professor Gregor Kasprian. He is from the Department of Biomedical Imaging and Image-Guided Therapy at the Medical University of Vienna. And he is a world expert and world leader in brain imaging, particularly of early fetal life. I'm going to hand it over to Gregor now. Take it away, Gregor, and then I will come in and give part of the talk. So.
Thanks a lot, Linda. Well, it's a real pleasure to be here and present. It's really unfortunate that I cannot be here in person in Texas. I personally have a history with Texas. I spent 1.5 years at the Baylor College of Medicine in Houston and was also working there as a research fellow. And it's really a pity because I really like the country and it would have been nice to refresh some memories. But however, it is what it is. I would like to introduce myself. I'm a radiologist, a neuroradiologist, and I'm working with imaging, and would like to share with you some thoughts on how we can diagnose the condition of callosal agenesis and dysgenesis, and would like to complement that with the expertise of Linda Richards in order to come to a point where you get a complete understanding of this condition. So we have to be aware that seeing the corpus callosum is not something very natural. You have to be aware that in the 16th century, the people were starting to appreciate that there is a structure between our two brain hemispheres, which is connecting both. Leonardo da Vinci was more focusing on the ventricles, so what is within the brain tissue, the brain water, basically these water-rich compartments. And at the same time, around the 16th century, anatomists like Andreas Vesalius made the first drawings of this structure. So this was the first time when people even tried to realize that there is something in our brain which is connecting the right and left side of the brain. Then it took us more than 500 years to really be aware that we can image and visualize this structure in vivo. And there we have two very eminent methods. One was computed tomography, which was introduced in 1978, and magnetic resonance imaging, with the first works on that and the clinical introduction in 1981. So be aware of the fact that we have a long time from the discovery of this structure on an anatomical basis to the state of the art of imaging. So the diagnosis basically in a living human being is possible since the late 70s and early 80s. So it's very, very recent, and we are really in a kind of revolution regarding this condition. So these are the first CT images of a subject with callosal agenesis. And you can see the quality of the CT here in 1982 was not really excellent. But what the early neuroradiologists at that time were able to appreciate was that the ventricles, these are the fluid and water-filled spaces within the brain, show a very distinct shape. If you look at that on an axial plane, which means you cut through the head in a transverse manner, you can see that these ventricles display a teardrop shape. So they are slim more anteriorly and more wider posteriorly. And that was one of these very early signs to diagnose the condition. So it was not even done by describing the corpus callosum, the absence of the corpus callosum itself, but more the configuration of the brain, which allowed neuroradiologists to describe the condition. These are the first MRI images, and you can see the image quality improved a lot, even within the 80s. This was 1985 on the left, and then 1988. Within 3 years, there were huge leaps in terms of tissue resolution. On the left, there is an image where you can see that there is a part of the corpus callosum present, and the rest is not present. This was a case of a Chiari-2 child with a open spina bifida and associated corpus callosum anomaly. And this was an image with a really, really better resolution. And you can see, again, the form of the ventricles, the inner brain spaces, is definitely different in a subject with corpus callosum agenesis. So it's not only that the corpus callosum itself is missing here in the midline, but it's particularly this very specific and peculiar shape of the corpus callosum, which makes this condition so special. And I mentioned already that some people thought that this looks like teardrops if you invert it, but as we are in Texas, we of course have to mention that this could look like bull horns or long horns maybe. So there is also the expression of bull horn configuration of the lateral ventricles. How does this look today? So here is an image of a normal human adult, a sagittal plane with a cut just in the middle, basically going through the nose to the back of the head, and an axial cut, which is transverse through the head. And this cut is exactly at the level of the corpus callosum. And if you cut this structure basically in the middle, you see this shape of the corpus callosum, as you can see it here. It's very bright on this T1 weighted sequence. Why? Because it is made out of white matter, and white matter is basically consisting of lipids, and lipids are bright on this special MR contrast. What we can do today is also we can use advanced imaging, and we are going to talk about this in a moment, like diffusion tensor imaging, to visualize the three-dimensional architecture of the white matter in vivo. This is also revolutionary. You have to imagine in previous and former times, pathologists had to dissect brains with a very special technique to see these white matter bundles. And now we have the possibility to really see those structures without any kind of administration of any contrast agent, just by using the properties of MRI. So this in blue here is the corpus callosum, and you can appreciate how complex and three-dimensional this structure is. And what most of the people are showing are these midline cuts through this three-dimensional structure, which gives you this very peculiar pattern of the corpus callosum. But in truth, it's a three-dimensional structure connecting both hemispheres. And now you see the image of a subject with callosal agenesis, and you see that there is the brainstem and the cerebellum. And here, where we have seen in the previous image this connection between the right and left hemisphere, there is simply no bright structure in the middle. So this structure is entirely missing here. And again, if you cut now the brain at this level, you would see that the ventricles appear widened in this in the posterior part, and here there is no connection between those both hemispheres. I go back here again to this image of the normal condition. You see here there is a connection, a broad connection between both hemispheres, and that is here absent. So these are the MRI features, but you have to be aware that there is a third method which allows us to get transectional images of the brain, of the human brain, and that is ultrasound. You can use ultrasound, of course, before birth, but also after birth in a neonate. These are images by a pediatric radiologist, and what you can do here is you can use the soft spot here at the front of the head in a baby to go through with ultrasound, put your ultrasound probe on that spot and look into the inside of the head, and by ultrasound also see that the corpus callosum here is not visible, and you can also see this form of the ventricles by ultrasound quite well. So this is the third method, which also gives you the opportunity to analyze and visualize the structure. And then, of course, is the question, can we see that prenatally? And here you have also to be aware that the history of that is a very short history. So Da Vinci was also thinking about how would life look before birth. People at that time did not really know what fetuses are doing inside the mother. So Da Vinci was one of the first, let's say, scientists thinking about the life before birth. And this is the first image by ultrasound of the head of a baby. And that was in 1961. And you can see that you basically cannot see much. You can see that you see the skull of the baby, basically the outer limits, the bone, but not really what's happening inside. At that time, you have to be aware also that the machine you need to produce this image was gigantic. So this was the transducer, what you see here. And this was called a diasonogram. And this was like an MR even machine. And here was the transducer put on the pregnant mother's womb, and then you could get this kind of image. Then MRI came around in 1983. We have this kind of image of a fetal brain with a malformation, a vascular malformation here indicated by the star. At that time, this image was revolutionary. However, I would have a hard time here to interpret the image. And what we see here is a more modern image of a so-called vein of Galen aneurysmal malformation, a vascular malformation in that baby. And this is the image contrast we are used to see today at 1.5 or 3 Tesla prenatal fetal MR examination. What you see here is a comparison between prenatal ultrasound of a world-renowned expert in that field and fetal MR. And I think you will appreciate that the details of a developing human brain can be appreciated really, really well. What you see here is that the corpus callosum connecting the both hemispheres can be excellently visualized by ultrasound through these cuts, which are coronal, meaning they go perpendicular to the level of the eyes and from front to back. And here, this is at different gestational stages. And here below, you see the MRI images at corresponding time points. And you can also appreciate that the MR contrast is also quite elegant. In some areas like here, laterally, it gives you a little bit cleaner contrast because MRI is not affected by shadowing of the human bones. So we can see better through the bones into the fetal head and brain of the baby. Again, here is the corpus callosum indicated. But we have also managed even to use techniques at prenatal stages to visualize the three-dimensional complexity of the corpus callosum. This is the technique called diffusion tensor imaging. This is, again, without any contrast agent, without radiation. You simply use MRI and try to use a technique called diffusion to see how the water is moving in the brain. And water can move freely or is restricted. And it may be restricted by white matter bundles. And so you can reconstruct this white matter through measuring water diffusion. And by that, you can see the different parts of this basically two-dimensional structure if you cut through it, but in fact, three-dimensional structure, the corpus callosum, even at prenatal stages of life. So let's go to the main topic of our meeting, the problem of a callosal disorder. So again, here, an image of a normal subject. This is the corpus callosum. There are different parts. Anatomists have realized that some parts are looking in a very peculiar fashion. So this is called rostrum, which means the beak of a bird or something. It's like in the very anterior portion here. Then here, that's the knee of the corpus callosum, or also called genu. That is Latin for knee. This is the body. This is the main kind of part. This is in Latin truncus, and here, or trunk. And here we have the splenium, which is the most posterior part of the corpus callosum. And it's also a large thickening here, connecting mainly the posterior parts of the human brain. So this shape you have to keep in mind whenever you look and want to assess the corpus callosum and its integrity and completeness. So when you again look at the subject with callosal agenesis, you can see that there is no corpus callosum at all here. You not even here in the rostrum area, you see nothing which is really resembling the normal condition. So in this case, we can say that we look at a person with complete agenesis of the corpus callosum. There is no corpus callosum seen at all. However, there are cases, and in many cases, you find something in between the hemispheres, which is connecting the right and the left hemisphere, which is by far not as thick and typically looking as in a normal subject, which is rather small, but it also does not show this typical configuration. So it's abnormally looking and it's too small. So this condition we call partial agenesis of the corpus callosum. Why partial? Because some parts of the corpus callosum are missing, others are there. And just to also explain this term, partial agenesis and hypogenesis of the corpus callosum can be used interchangeably. Genesis means basically that something is developing and it is growing. And for that reason, if something is growing too little, it says hypo. So if something is too little or less, so underdevelopment of the corpus callosum. Then we have a situation where the corpus callosum is there in all parts. You see here again the genu, the rostrum, genu, truncus, and splenium. But what you can see is that the thickness compared to this man here, the thickness of the corpus callosum is by far not as what we can see here. So this is a hypoplasia, which means the corpus callosum has formed in all parts, but has not reached its final thickness, mainly maybe because there are too little white matter fibers connecting here, and the white matter of the brain in the hemispheres is not evolved to a proper number of connections, and therefore this midline structure is thinner than we would expect, so hypoplasia. Okay, this is again a you can all notice that again. These are the typical images of a corpus callosum agenesis case, and here you again can see this bullhorn configuration of the lateral ventricles and the teardrop-shaped configuration here on an axial plane. What you can also appreciate is that the ventricles here in the front are very small, and in the back here are rather thick. And this is called, or a neuropathologist in the 60s, Jan Bender, was calling that colpocephally. And kolpos means in Greek something like a vessel or something where you can drink out of it. And he was referring to that shape that here the ventricles are larger in the posterior side. So we can nowadays even look at the development of those ventricles before birth, and we can basically use and analyze our baby images to see how the brain is growing and how it looks before birth in 3 dimensions. So here you see the work of my PhD student Marlene Stümpflen, who has segmented those brains, and you can wonderfully see how smooth the baby's brain compared to an adult brain is. And you can even then virtually dissect without doing any harm to mother or fetus, virtually look through all these anatomical structures, and you will finally also see then the ventricular system, and this is how a normal ventricular system of a baby looks like. So these anterior horns, the frontal horns, are rather thin. In this region, it's a little bit wider also physiologically, but not very wide. And you see that throughout development, different parts are growing at a different pace, basically. And you see, particularly also here in the posterior part, during our prenatal development, these structures are expanding and earlier and then reducing in width. And now I would like to show you that the brain of a baby with callosal agenesis is already different before birth, clearly different. So this is the surface. This is not an optimal reconstruction, but just look at the ventricles. These are the ventricles in a normal subject. And this is a fetus, a baby with callosal agenesis. And you see that here, in this part here, in this 3-dimensional reconstruction, the ventricles are really, really wide. So this is what we call colpocephally, and we have done some analysis, and what we found is that that this is mainly due to the fact that the brain tissue is thinner in that region. So I told you that you can use this technique to visualize the pathways in 3 dimensions. I will not go into the details here. I just simply said in each point of our image, we can identify the main orientation where water is moving in the human brain. And by that, and the computer algorithm, we construct these kind of fibers. We do not know of those fibers, where they begin and where they end. So the beginning and the end can be interchanged. What you also see is that this technique allows you a color coding of the white matter, and this color code tells us where the fibers are actually going. And just keep maybe in the back of your head that the red fibers are connecting from right to left and left to right, and the green fibers are going from front to back. And of course, the corpus callosum is a right-left structure, so it really contains a lot of fibers going from right to left. And this now is the brain of a subject with callosal agenesis, and you see here, there is nothing red in between the hemispheres here. But what you see is there is a green structure here, and these are bundles of which tried to form the corpus callosum but could not move over to the other side. And now they are running from front to back, and they are called the bundles of Probst, because Moritz Probst was a psychiatrist describing this for the first time, and that's how this looks like. So actually, callosal agenesis is a misnomer. It means that it is absent, but actually the fibers which are forming the corpus callosum, they are there. They are just running a different direction and giving this typical impression. Nowadays, we can calculate a lot on basis of images, and we can see also how different brain areas are connected, and that's what we call connectome, but I will not, for the sake of time, go into that further. What I would like to tell you as well is, and show you the following case. This is a 5-year-old with a problem of the right hemisphere with epilepsy coming from this right hemisphere. It's called polymicrogyria. And you see also here the corpus callosum is very thick, and the shape is basically a little bit abnormal compared to what we have seen before. Sometimes in this situation, the only way to treat this patient properly, to treat the seizures of this patient properly, is to remove the corpus callosum in the midline, to kind of cut through it surgically. And by that you can help the patient that the seizures get less. However, of course, you are cutting through a main structure of the human brain. And of course, this will change the way the brain looks like. So here is this callosal agenesis subject. Here is a normal subject. You see this reddish color, right, left color, fibers. And now if you cut that in the middle, you see that this changes a little bit. The red goes away, but these green bundles you see here in the callosal agenesis subject is still not present here. So there's a huge difference between a subject where you secondary cut through the corpus callosum and a subject where the corpus callosum simply did not form in a proper way. These brains are totally different from an imaging perspective. And of course, you can virtually dissect this also prenatally, but I will not go into that, because the really important thing here is to understand why are these brains of persons with callosal agenesis so different. And in order to understand this, you also need to understand the origins of the corpus callosum. And here I definitely need some help from Linda.
Okay. All right. Well, thank you very much, Gregor. If you can do the slides from there, that'd be terrific. We actually only have 10 more minutes for this talk, so we will have to speed it up a little bit. Otherwise, we won't have any time for anybody to ask any questions. Can I get rid of that? Yeah. Okay, so what is the cause of complete agenesis of the corpus callosum? The key here is actually this structure which sits below the corpus callosum, and has another anatomical name called the septum pellucidum, or the septum. So what you can see here, this is the neurotypical on this side, and the person with corpus callosum agenesis, no crossing, on this side. So this is a view this way, as Gregor had said, and the bottom ones are views this way. It's the same brain. And so what you can see is the corpus callosum would normally be crossing here, but it's not. And so, yes, they're Probst bundles. And so what you can see, though, is you see these two little bits of tissue here. That is the septum, which is now open, compared to over here, which is closed. So how does it get closed? So next slide. Right, so the key here is this thing called the interhemispheric fissure. So that's the division between the two hemispheres. And essentially, in early brain development, those are separated and they have to knit together, and a bridge is formed by some other cells in the brain. They're called glial cells that come from the middle of the brain, and they migrate forward and knit together the two hemispheres. And when they do that, they provide this bridge for the callosal fibers to actually grow across from one side, across the midline, this fissure, to get to the other side and then find their right target in the other hemisphere. Next slide. Can you change the slide? Yep. So what you can see on the left is a mouse brain. This is where I said, like, mice actually have brains that are not that dissimilar in early stages. And if you have a look over here, this is the mouse brain. You're looking head on to the mouse brain. This is a scanning electron micrograph. And what you can see is the interhemispheric fissure here in a mouse is also separated. And this is a human brain. This is what a human brain looks like at 13 gestational weeks. And you can see this is the fissure there. All right, next. Okay, so if we look at a human brain at gestational week 13, 14, or 15, you can actually stain the brain. If you slice it, so this is actual, take the brain out and slice it. So this is what we've done here. And you can see the glia are stained here. And this is where the brain is becoming fused together. All right. Next slide. And so you can think about this like a zipper. So early in development, the zipper's open. And then between 12 and 16 weeks of gestation, the zipper is closing. And during that time, the fibers start really crossing. All right, next slide, I think might be yours. Okay, this is an MRI of a fetal brain. It's one brain, but you can see at different parts. Here's where things are very much fused already, and here's where things are still open, not fused. You can see the fissure in between here. And so this occurs actually from the bottom part to the top, and from back to front, but in the very front of the brain. So it's a very precise anatomical reorganization of the structure that occurs. Next slide. Yes, and we can study this in mice. So we can look at this in a mouse. This is now a mouse brain, sliced this way. And what we're doing is staining it for the glia and also for the fissure. So the fissure, this IHF, is the interhemispheric fissure, and the purple are the glia. So you can see it starts out, these are different stages, embryonic day 14 in a mouse, 15, 16, and 17. So across development, you can see it's open. So we're looking this way, down on top. And this is the front of the brain, and this is the back of the brain. And you see these glia form here, and they begin to migrate forward, and they knit together the two hemispheres. So we know this is how this occurs because we can see it, we can measure it, and many genes are involved in forming the bridge, right? So next slide. And we know that the callosal axons, so here now we've labeled in the mouse the callosal axons as they're coming across the bridge, and we know they won't cross here where the green is, where there's a bit of tissue in between, but it's not the right tissue. They only cross where the bridge is. They have to have the bridge. They don't have the bridge, can't cross. They grow back and back, forwards and backwards. Okay, next. Okay, we won't go over that. Let's just keep going. And back to you, Gregor. Just remember, we only have now 3 minutes, so you may want to pick a couple of things, and we'll skip the second part that I was going to do so that we can have some questions from the audience. Okay?
Okay. I hope you can hear me now. So one question people might wonder is, I have mentioned the term colpocephally and referred to that multiple times, and have said that the ventricular system of persons with callosal agenesis looks different than a normal situation. But what is the difference now between this colpocephaly and the hydrocephalus? And this question comes, of course, from you who know already that the water within the brain is produced by a structure which is called choroid plexus in here. And this water basically travels through this ventricular system from basically up here down to here in the posterior fossa, and it leaves basically the inside of the brain here through very little openings down here. So you can imagine that if one of those openings here is obstructed and closed, the water produced by this choroid plexus in the inside of the brain cannot leave the inside. And this is an example. So this is a baby where exactly in this region hemorrhage, a bleeding occurred, and this opening was obstructed. And as a consequence, the water inside there could not leave and was accumulating within the ventricular system and was basically pushing the brain aside and the cerebellum up. And this was the image which was generated, and this was the image at the level of the ventricles here. So please note that the frontal horns, this frontal part of the ventricles, is much, much wider than we would see it in a subject with callosal agenesis. And also the configuration of the ventricles is different from what we used to see here. So in callosal agenesis, there is no problem of circulation of this brain fluid, of this brain water. This is not a problem at all. It's just the configuration of the brain around that ventricle which determines this peculiar and specific shape, this colpocephaly. I will also jump over that. We have just prepared with Linder some considerations why these brains are looking somewhat different, and that has to do with migration. I will also go over that in order to have time to discuss, but just shortly, that in order to have a proper brain, nerve cells have to divide and move to the proper spot. And if they can't do that, you have something like a malformation. So, which means in addition to the absence of the corpus callosum, also a part of the brain is not developing properly. And this was the case here in this baby, where you see this dark line here on the one side of the brain occurring. This is a very decent finding at prenatal stages of development, associated with almost no abnormality on the surface. But if you look at this child postnatally at the age of 12 months, you can see here that this line has grown into a structure which we call double cortex. So, which means the gray matter of the brain is outside here and should always be outside, but there is some gray matter in the inside, within the white matter, and these are non-migrated neurons which did not move out to the cortex. So this is a malformation, and this is a condition which is often and frequently associated with callosal agenesis. And if we have time in the discussion, we can a little bit talk about why that could be. But just keep in mind, developmental brain disorders often there is a link between callosal growth and also cortical development and the development of the rest of the brain. So with that, I would like to thank for your attention, and I think we are going to the discussion.
Right. Absolutely. Thank you so much, Gregor. We have a couple of minutes. Yes, let's say thanks to Gregor. We have a couple of minutes for questions, and then we're going to have the genetics team come up. So that's why I wanted to keep us on time. And sorry, we went through that quite quickly, but I'll be around for a couple of days, so I can go through it more slowly with you if you need to. So does anybody have a question I would like to ask regarding the imaging? Yeah. So Gregor, I'm not sure if you heard the question, but let me repeat it. So a person in the audience had a brain bleed when they were during gestation. And the question is, could that have caused the corpus callosum disorder? Do you want to answer that? Oh, yeah.
So may you repeat what could have caused the callosal agenesis?
Could a bleed in the brain have caused?
A bleed.
Yes.
That's an excellent question. You know, in development, it's always important. Timing is always important, and it really matters when a hemorrhage is occurring. So if a hemorrhage occurs when the corpus callosum has fully formed, it will, of course, lead to a defect in the corpus callosum. So it's like a missing piece there because it was destroyed there, but it has developed before that. So when the hemorrhage occurs in a very early time point, and these are the time points Linda was showing where these axons are starting to cross over, and we would have a hemorrhage at these early time points, for instance, due to an infection, then these axons would not be able to cross over, and then this could be also a so-called acquired origin, non-genetic, but acquired origin of callosal agenesis. Yes. So it occurs, but we believe it's not that frequent.
We had some questions on Zoom. Okay, let me give you this one. Can you see Blake's pouch cyst on the MRI fetal picture? Have you had experience with the combo Blake's pouch cyst and ACC? Can the cyst be the reason of absence of the corpus callosum?
Thank you for this question. So I think the term Blake's pouch is already a very special term. You have to be aware that I showed you previously, and I think I'm still sharing my screen. So if I go to this image here, right here, you see that this is the fourth ventricle, and it's full with fluid. And in situations where this ventricle, these openings are opening too late during development, there may be a cystic dilatation of this fourth ventricle, and that is called a Blake's pouch cyst. And by definition, Blake's pouch cysts should not go along with any cerebellar malformation. So the association between Blake's pouch cyst and callosal agenesis is basically possible because it's an association. You know, there is everything can go together somehow, and also by chance. But I'm not aware of a real causative connection here. Posterior fossa malformations have a higher incidence and go along often with callosal agenesis, but I would a little bit doubt the diagnosis of a simple Blake's pouch cyst, which we rather refer to a physiological phenomenon in this condition of callosal agenesis.
A related question that people might have is about cysts in general. And definitely if a cyst forms at the midline, at the right stage of development, when the corpus callosum is forming, that can cause a corpus callosum disorder. And that occurs in Aicardi syndrome, for example. And that was one of the things that I asked Gregor to skip over because we were running out of time. So, and Elliot. Big pardon? Right. Okay. And another reason is the formation of something called a lipoma. So this is a kind of fatty substance that occurs at the midline of the brain, and it looks like a tumor, but it's actually a fatty kind of substance that doesn't continue to grow, and that blocks the bridge. So similarly, if the cyst is in that place, it blocks the formation of the bridge, then you get a corpus callosum disorder. Yeah, so you can see lipoma on MRI and by ultrasound. Yep. Yeah. So for people on Zoom, the question is, can you have a lipoma in the brain at one stage and then later form lipomas in other areas of the body? And I'm going to ask Elliot to answer that.
Well, it is a very interesting question.
Gregor is going to answer it. We got a lot of experts here. Go for it, Gregor.
Sorry, I will leave it actually then to you, because I have seen cases with callosal lipoma, and to me, I'm not aware of an association between this lipoma. I can only tell you from the prenatal perspective as well, that fat in a baby does not always look the same on imaging. So initially, a callosal lipoma may appear as a thickened corpus callosum, and later at later stages of imaging, like just before birth, it shows then the typical characteristics of fat. And then also lipoma, because they are fat cells, they are basically getting fatter and getting more voluminous at later stages of imaging. If there is a genetic association with subcutaneous lipoma, I'm not immediately aware of that, but here maybe Linda or somebody from the geneticist can help me.
Yep, so Elliot was about to make a comment. Okay, so here is, like, we know nothing about the genetics of lipoma currently. Yeah. I am very interested in lipomas, how they form, what kind of tissue forms them. Like, they're called fat cells. You know, that's not a very specific term. So how do they form? Where do they come from? Why do they form? Is there a genetic basis to this? We don't know. We absolutely don't know. It's a really good research question, though, because if we knew, then we would be able to tell you why and how and what does it mean. And I think this will be the last question because we need to move over to the genetics group. Go ahead. Got it. All right. So let me answer the first question because I confused you before when I said it forms from back to front. It only forms from back to front in the very front part. The corpus callosum forms from front to back overall. The front part forms first, then the middle, then the back, 100%. But in this area where the bridge forms, that only forms in the front part, and that forms from front to back, but only in this little bit in the front. So that's that. The second question was, is there a good clinical reason to continue to have imaging done? Gregor. At different times during life. Sorry, that was the question. Can you take that one?
Yes, I just acoustically, is there a clinical reason to?
Oh, to have multiple MRIs done at different times
Times.
during life. So you've got your diagnosis. Should you go back and have another scan done at 5 years, 10 years, 15, you know, as an adult, and, you know, maybe have some of the more sophisticated imaging done?
Well, let me start with that I have shown you that we can do imaging already before birth, brain imaging before birth. There, the diagnosis is very critical, and you want to know if there are other things associated with the condition, if it's isolated or if a brain malformation goes with it, if there's a risk for epilepsy. So prenatally, it might be that you have, let's say, multiple imagings, one at around 20 weeks where the diagnosis is made, and then later on in the third trimester, just to get a more complete picture. But if, let's say, the baby is born and there's a live human subject, we at our center, we do not kind of push for imaging, because all we need to know is already done actually before birth. We only would go for imaging if the subject says, well, I'm interested in that for scientific reasons, but the main reason would be if there is a symptom, the patient is presenting with a symptom which was not expected by the previous imaging. So that counts basically for all of us. If we have a symptom, suddenly an epileptic seizure or headaches, or the head circumference, the size of the head changes suddenly, well then we of course need, that's not different from anybody else, another set of images to identify the reason for that. So I have seen persons with callosal agenesis having imaging at 60 years because they had a stroke. So the brain of those subjects is not different than any other brain, and of course you need another imaging session, but not because you have the callosal agenesis. That is basically set very early.
All right. Thank you so much, Gregor. We're going to move on to our genetics team now, and thank you all of you for asking questions.